Alright, so hello everyone. Today we are going to understand hemoglobin electrophoresis in a very simple and logical way. So by the end of this session, you should be able to look at the hemoglobin electrophoresis pattern and quickly identify whether it is the normal adult, it is the normal newborn, the sickle cell trait, and the rest of the types. We need to differentiate that after looking up to the hemoglobin electrophoresis. So the very first thing that we have to understand what is hemoglobin electrophoresis, right? So this is one of the lab techniques. This is one of the lab techniques used to separate the different types of hemoglobin based on their electrical charge, whether that is the negative or the positive one. So one of the major key idea behind all of this concept is that the different hemoglobins have the different charges. So when we place them in any of the electrical field, they do not just move all, they do not move the same distance. For example, I repeat this one, that whenever we do check out the electrical charge of the different types of the hemoglobin, and let's say because different hemoglobins or the different types of the hemoglobins, they do have the different charges. So when we place them in the electrical field like this one, all of them, they do not move to the same distance. Some of them move farther and some of them move less, and some remain closer to the starting point. So you have to remember it out that the hemoglobin electrophoresis separates the hemoglobin types according to the charge, right? So before we looking at the pattern, let's understand the overall setup. What happens during the electrophoresis? You have to remember of this one, that in this method, the hemoglobin is placed on a gel. Okay, the hemoglobin, it is placed in on the gel, right? And after that, any of the electrical current is applied to that one. Okay, and the hemoglobin starts near the origin, which is close to the negative electrode. It means that this is the origin part and this is near to the negative charge or the cathode region. Okay, and from there it starts migrating or shifting towards the anode side or the positive side, right? So this is very important because the more negative a hemoglobin type is, the more strongly it is attracted towards the positive anode and it travels. Okay, because the more positive it is, the less far it goes, so it stays closer to the cathode, right? The opposite one. Okay, so the whole overall story, it depends upon the charge. Now we have to remember it out the order of the migration, which is absolutely very high yield. Okay, very first thing I have to remember it out the sequence. We do have the hemoglobin A, right? We do have the hemoglobin F. We do have the hemoglobin S, and we do also have the hemoglobin C, right? So you can just look up to these 4 months first, and the order of the movement, right, or the order of the migration, it is just like this one. And out of this one, if you just remember this part, right, you're going to remember it out the migration format of the different types of the hemoglobin. So it according to this one, the hemoglobin A moves the farthest, right? This is the one which moves the farthest, right? Like moving so much away from everyone. And we can say that after that, the order comes the hemoglobin F, then the S, and then at the last, that is the C. It means that the hemoglobin C, it moves the least. Okay. Now you see that the C, F, C, S, F, and A over here, right? So you see that from the origin, which is the one which is moving the farthest, that is the A, and which is the one which is moving the least, that is the C. That is why you have to remember it out this specific sequence. Okay. So if you're moving from cathode towards the anode, like from cathode to the anode, so it would definitely be the CSFA, right? You have to remember it out. This is one of the classic order, which is also being given out over here, that HBC is closest to the cathode. You see that this is the cathode, so at the hemoglobin C, this is the closer one to the cathode, and the hemoglobin A, this is closest to the anode, right? Do remember it out, these ones. Right, so and that is why this is one of the mnemonic that you can remember about, that a fads and a close can't go far, right? So you have to now cover up to one of the questions, that why do hemoglobin S and hemoglobin C migrate less than hemoglobin A. The question is that why this hemoglobin A, it moves the farthest if we compare it with the hemoglobin C and with the hemoglobin S. So now let's understand one of the molecular reason behind that. This is where most of the students memorize without understanding. So hemoglobin A is the normal adult hemoglobin. Do remember that. This is the normal adult hemoglobin. Okay, do remember this one. And in the hemoglobin S and the hemoglobin C, there are the mutations in the beta chain. Okay, in the hemoglobin S, right, and in the hemoglobin C, we do have the mutations in the beta chains, right? So in the hemoglobin S, the glutamic acid is replaced by valine. Okay, do remember that the glutamic acid, it is replaced by valine, right? And the glutamic acid is negatively charged, while valine, this is negatively charged, but this is the neutral one, not having any charge. So hemoglobin S loses some negative charge compared with the hemoglobin A, right? And so because of this one, because it is now less negative, it is less strongly attracted towards the positive anode, so it does not migrate as far as the hemoglobin A. And in the hemoglobin C, in HbC, this was for the HbS, right? And for the hemoglobin C, you have to remember it out that the glutamic acid is replaced by lysine. The glutamic acid is replaced by lysine. So lysine is positively charged in the hemoglobin C. So the hemoglobin C becomes even more positive compared with the hemoglobin A. That means it migrates even less, and therefore it says that it stays closest to the cathode one. Okay, to remember this one. So the logic that you have to remember it out, that hemoglobin A, this is the most negative among all of these ones. That is why it is moving most farthest as compared to the other ones, right? And this is more positive than hemoglobin A, so it moves the least, right? This is the biochemical basis of this question, that you can remember it out. And it is not just a random movement, it is all because of the charge differences caused by the amino acid substitution, right? I have already told you that in hemoglobin C, glutamic acid is replaced by lysine, right? And in hemoglobin S, that is glutamic acid replaced by valine, right? Very high yield to remember now. Now, the next question that's been arises is that how to read the electrophoresis chart, right? So now just see that. You see that when the reading hemoglobin electrophoresis, the first thing you should ask that where are the bands, right? And the second question, that how many major bands are present. So each band tells you which hemoglobin type is being present. So now let's go pattern by pattern. So over here, this is the chart that you're actually looking at. Okay, the first one, this is AA, right? Means it mentions of the normal adult. You have to remember it out. In the normal adult, the main hemoglobin is hemoglobin A. Okay. And when you see a strong band at the A position, whenever you see a strong band on the A position, right? Over here, you see that this is the A position. Now, this is very strong. This is wider. This is larger in the size. So whenever you see a stronger band at the A position, this means that this person has the normal adult hemoglobin. If you want to say it in the, let's say that if you wanted to say in the very other language or in a very simpler way, you can just simply say that when the electrophoresis, it shows only the A band, right? This represents a normal adult pattern, right? Do remember that. Now let's look up to the AF. This represents the normal newborn, because newborns still have the large amount of the fetal hemoglobin, okay? And they do also have some of the hemoglobin A, but largely the fetal hemoglobin, which is the hemoglobin F, okay? So in a normal newborn pattern, you will see what? You will see the HbF, okay? You will also see the HbF, and you will also look up to specifically the HbA, right? So the both of them, and that is why it's been written as A and F. Yeah, that is why it's been written off as A and F, right? So the fetal hemoglobin, it starts actually decreasing gradually after the birth, while the adult hemoglobin starts increasing, right? It increases. So you have to remember it out. If you look out the both F, right? You see this band and you see this band over here. If you see both of them, that is the AF, the normal newborn. Okay. Now another pattern, that is AS, right, representing the sickle cell trait. So over here you have to remember it out that the one normal beta globin gene producing hemoglobin A pattern, and one sickle beta globin gene producing the hemoglobin S, right, over here. And one gene is producing the normal one, but another gene that is producing actually the S1. So the both HbA and HbS, they represents the A and S. All right, and electrophoresis, we are going to observe one band on the S side, another band on the A side. This is the sickle cell trait, but not a disease. This is important because these people are usually the carriers. These are still the carriers. They usually do not have the full-blown sickle cell disease, okay? Although under certain conditions, they may have the complications. So the key interpretation is that A plus S to remember the sickle cell trait, and they are still the carriers, not the disease, okay? Then we do have the SS, right? The both of them, they do have the absence of the A, right? But they do have the large band of the large band on the S side, right? Over here, as you can see that the patient doesn't have any sort of the normal hemoglobin A. Instead, they do have the major hemoglobin present, that is the hemoglobin S, right? So that is why it is showing you a strong S band without hemoglobin A. And this pattern clearly indicates the sickle cell diseases, right? Have to remember it all. And you have to remember that over here, how to differentiate that if we do have present slightly the A band and the S band, that is the sickle cell trait. And if you do not have any sort of the A hemoglobin, right, that is only an and having the large band of the hemoglobin S, then that is the sickle cell disease. So the next one that you're going to look up, that we do have one normal beta globin gene and one HBC gene. So the patient does have again what? The trait, right? The A and the C1. This gives the both of the bands to us. HPC migrates the least and its band is closest to the cathode. So the pattern still is the trait, but not the full disease. But what if we do have the full band, right? That is completely what the HPC disease one, right? You have to remember it out. Okay, and then we do have the SC, right? Absence of the A, but the presence of the both, the C and the S band, right? And over here, this is a disease state and not anymore the trait or the carrier state. So clinically, the HBSC, right? It can be significant and may cause the sickling related problems, although often the severity differs from the classic HBSS diseases. So if you see no HbA but both S, so that is the disease, that is the hemoglobin SC, right? Over here you can see and find out the different interpretations for that one, right? I hope so that this is important and this is easier to now understand, right? And read up this all one carefully. This glutamic acid minus means that the absence, the presence means this positive means the presence for that. uh, some of the basic questions that they usually do wanted to cover up, like the chart. The understanding of the chart is important. Why this A is the hemoglobin A is farthest, but the hemoglobin C is the closest one. So you have to think of that all carefully. Thank you very much and look up to another session.